Hello everyone, welcome to lecture 4. Today we're going to be talking about the pattern recognition receptors and their outcomes. So again, just a quick look at this. We're going to be looking at all these different figures today. All these different receptors. We're kind of going to look at how they're all activated and how they work. Take a deep look at TLRs. And yeah, just keep that in mind. So just a quick recap. So last time we looked at complement and its 3 different pathways. So just to keep it relevant, complement is not, it's actually quite topical because there's a lot of research saying that SARS-CoV-2 or COVID-19 is highly involved with the complement system. So here we can say hijacks complement, so we can have overactivation, coagulopathy, so it can be involved in too much coagulation. Here we can see again it's involved in the severe COVID-19 ARDS patients and can be involved in multi-organ complement deposition. So today we're first going to start with a case study of CGD, also known as chronic granulomatous disease. So here we can see CGD is a primary immunodeficiency disease. And here you can see here granuloma. Here's a picture. You can see it will be key in this formation. You can see them here. So chronic granulomatous disease. So in normal phagocytosis, which we can see in A, we have damage to ingested bacteria is carried out in the fecal lysosomes by toxic oxygen intermediates through the neutrophil extracellular trap formation by neutrophils. But in CGD, as you can see in B, a key enzyme needed for the respiratory burst is defective and causes impaired killing of microbes. This enzyme is NADPH oxidase, aka phagocyte oxidase. So in phagocytic stimuli, there's the assembly of the NADPH oxidase complex, which we can see here in this figure, in the phagosome membrane. In CGD, the most common defects are to the GP91 subunit, so the enzyme cannot be assembled properly. So here we can see, without NADPH, we can't have this reaction, and we don't have our reaction oxygen intermediates. So again, CGD illustrates the importance of phagocyte activation. So activated phagocytes are more effective at killing pathogens due to the reactive oxygen species production in the respiratory burst. So macrophages and neutrophils can be activated by contact with bacterial products such as FMET and LPS, and certain complement components like C5a, and certain cytokines like interferon gamma. We can see that reaction oxygen species are generated in a respiratory burst and can kill microbes. The subunits of NADPH oxidase or the phagocyte oxidase assemble in the phagosome membrane and generate the ROS. Neutrophil extracellular trap or net formation in neutrophils only occurs after reaction oxygen species generation occurs. So in CGD, we have defective net formation and defective respiratory burst. So here again, we can see the reactive oxygen intermediates, as we discussed, superoxide, hydroxyl, hydrogen peroxide, and hypochlorite, nitric oxide, and etc. So, as we discussed in the last chapter, we can use NBT or nitro blue test to view these cells. So, and another way to measure respiratory burst activity is through the dye, like I said, dihydroamine. So first we need a stimulus, which we use formyl mesarate acetate or PMA, which is added. This stimulates phagocytosis to produce superoxide radicals and reduces the dye DHR and miromidine, which is highly fluorescent. So this is our way to view it, essentially. So something to keep in mind is granulomas. Why did they form? So here we can see granuloma formation. In a typical bacteria, this occurs with Mycobacterium. And here's a micrograph. So what types of infections are problematic in CGD? So when we don't have respiratory bursts and nets, people with CGD suffer from an increase in bacterial and fungal infections, but not viral infections. So what does this tell us about innate immunity? So here's some of the common things. We can see Staphylococcus aureus, which we've talked about before, Salmonella, Mycobacterium, and more. Essentially, microbes which would usually be killed by activated phagocytes are the ones that will get us, cause us to have infections. So pathogen recognition and tissue damage initiate innate immunity, and they induce an inflammatory response. So our cytokines, which are produced by our macrophages, cause dilation of small blood vessels. The leukocytes move to the periphery of the blood vessels as a result of increased expression of adhesion molecules, and we'll discuss these later, and some of them include ICAM, intracellular adhesion molecule, but anyways, we'll get to those later. And then the leukocytes extravasate to the site of infection. So macrophages and neutrophils interact to respond to pathogens and generate an immune response. But how do they recognize these pathogens? They do so through pattern recognition receptors, which we'll talk about now. So, the mechanism recognition. We have our PRRs. PRRs recognize the PAMPs we discussed earlier, our pathogen-associated molecular patterns, and also MAMPs and DAMPs, which are damage-associated. So these are when cells are stressed, so our cells are somehow encounter damage or under stress, these will be expressed. And we also have our microbe-associated patterns. So we have our different types of PRRs. So first, we got our phagocytic PRRs, which recognize PAMPs on pathogens and trigger uptake. We have our secreted or free PRRs, which are involved in opsonization and complement activation. These include MBL, like mannose binding lectin, which can bind to bacteria, which activates the complement cascade in the lectin pathway and promotes opsonization. We have the signal transducing PRRs, which signal the presence of microbes and induce signal transduction pathways and cause a response. These include membrane-bound PRRs called TLRs or toll-like receptors, which are transmembrane proteins, but not all they're in the same membrane though, and intracellular or cytoplasmic PRRs, which include the NOD-like receptors, which are cytosolic. So we can see here that TLRs bind to molecules with specific motifs expressed by various microorganisms. So it is hypothesized but strongly suggested that these kind of develop through evolutionary techniques, that we develop responses to things that were essentially always going to be foreign. So here we can see stuff like LPS. We're never going to have that naturally. So we have TLRs designed to respond to certain things. As we can see here, we'll get to this in detail later, but just keep that in mind. And we can also have secreted PRRs like soluble MBL, which bind to bacterial polysaccharides and result in opsonization. So, phagocytic PRRs. These stimulate phagocytosis and intracellular killing of pathogens. They recognize PAMPs and MAMPs on pathogens and induce uptake by phagocytosis. Examples include our complement receptors, or CRs, that we've seen before. They involve the uptake of complement-coated microbes. And we also have our macrophage mannose receptor, which helps in the clearance of proteins with attached mannose in microbial and host-derived. We also have our scavenger receptors, which include bacterial cell wall components that detects bacterial cell wall components and high-density lipoproteins. So as we can see here, our macrophages have phagocytic receptors that bind to microbes and their components. Again, mannose, complement, even lipids, and our scavenger receptor, as discussed. And our dectin-1, also known as which detects glucose polymers or beta-glucans. So, signal transducing PRRs. A look at TLRs. So a lot of the rest of the lecture will be focused on these, and these are really important and also involved in COVID, as we'll talk about later. So here we can just see a TLR on a cell. We can see the sensing microbial products inside and outside of human cells by different TLRs. So we have different TLRs in different locations, and they all recognize different specific things. So again, some TLRs are cell surface associated, while some are intracellular, some are homodimers, and some are heterodimers. TLRs can sense a variety of microbial components. TLR binding results in a signal transduction and activation, which we'll go through later, of phagocytes and APCs. So each recognizes a different component of the bacterium or pathogen, and we'll discuss that later. So if you guys want a really, really detailed look at the TLR pathway and even the NOD pathways, this is a really great website, although it's like they use it to sell product because eventually they have products that can pretty much detect everything within these pathways to some degree. But it allows you, it's an interactive kind of thing. You can go through and follow, let's say I just want to follow TLR4. So you just follow TLR4 all the way through, or you can have them all activated and you can kind of piece it together one by one with the animations. So it's a good resource to check out. So, what do TLRs see on bacteria? So here we can get more specific. So we can see TLR2 looks at lipoprotein, but mainly you can be considered cell wall components. While TLR4 which is probably one of the more talked about ones, and you'll see it in other types of research too, is response to lipopolysaccharide, which is very common in gram-negative. Then we have TLR5, which binds to flagellin on bacteria and TLR9, which binds to bacterial DNA or CpG motifs. And here we can also see TLR2 binding to another different type of cell wall component here as well, and TLR5. So here we have this filled in. So TLR2, again, cell wall components, including peptidoglycan and liponucleic acid. TLR3 can sense double-stranded DNA from viruses. So we can see here if they get into the cell, we can detect it and try and we'll have our pathways that will try and destroy it. TLR4, again, key molecule here, lipopolysaccharide in gram-negative bacteria. TLR5, flagellin on bacteria or viruses. And then TLR9, which binds to bacterial DNA or CpG motifs. So essentially, this is the go-to figure here if you just want to get a sense of what each TLR does. So again, we go from 1 to 11. This is what's, as you can see, TLR is mouse only. But again, even here, unknown, so not everything is known. But I would say there's a good amount of information from 1 to 9. A variety of TLRs allow for recognition of a wide range of potential pathogens. TLRs 1, 2, and 6 bind to bacteria, mainly gram-positive, where mycobacteria and yeast. And then TLRs 3, 7, 8, and 9 recognize viral and bacterial nucleic acids. And then we have TLR4, specific for LPS. So TLR structures and their key features. So TLRs are horseshoe-shaped transmembrane proteins, as you can see here. Extracellular pathogen, they have an extracellular pathogen recognition domain. They have a cytoplasmic signaling domain, as we can see here. They also have pathogen recognition domain, has repeated leucine-rich repeats, so that's each one of these. These are LRRs. And the TLRs are vary in number of the repeats, so each TLR has a different number. We also have a cytoplasmic signaling domain, or the TIR domain, as we see here, which is called the toll interleukin receptor domain. The TIR domains are found in TLRs and in the interleukin-1 receptor. That's kind of where it gets its name. And IL-1 is a pro-inflammatory cytokine, so just keep that in mind. TLR stimulation by microbial components early in infection can induce innate activity, and this sets the stage for the adaptive response. Some TLRs form homodimers like TLR4 and 3, and some are heterodimers like TLR1, and only 9 leucine repeats per TLR are shown here for simplicity, but there's usually 18 to 25, but it varies. So they can also vary in location. So here we can see 2, 6, 2, 1, 5, 4 on the plasma membrane, where we can see 3, 7, 8, 9, which you can see here are involved in DNA, RNA, and DNA RNA recognition are in the endosome. Some are located to detect extracellular PAMPs, like cell structural components and flagellin, and others are located to detect intracellular PAMPs, usually viral or bacterial DNA. So certain TLRs are usually on the cell surface, as we said, TLR2, 4, and 5, which detect extracellular PAMPs, including cell wall components. Certain TLRs are intracellular and are anchored in the endosomal membranes, including TLR3, 7, 8, and 9, which sense intracellular PAMPs, including viral RNA and bacterial DNA. So this figure, we can see that extracellular domains of TLRs have binding sites for distinct PAMPs. When two TLRs recognize a PAMP, such as a lipopeptide, the TLR dimerization is induced and brings a cytoplasmic toll IL-1 receptor, or TIR, domains together to initiate signaling. So here we can see they come together, binding to the PAMP, and we have our activation. So signal transduction in response to TLRs. So most TLRs share common signal transduction pathways. They activate gene transcription and result in cell activation and release pro-inflammatory activity, usually through cytokines. A common theme is the activation of NF-kappa B. So NF-kappa B is a transcription factor, normally held in the cytoplasm in an inactive state by I-kappa B, which is an inhibitory protein. Essentially, the goal of TLR signaling is to release NF-kappa B from I-kappa B so it can get to the nucleus and turn on transcription. So, ligand binding to the cell surface receptors recruits lipid rafts to the receptor complex, and this recruits signaling molecules, and this improves efficacy of signaling. Signaling processes involve adapter proteins, protein tyrosine kinases, or PTKs, and protein tyrosine phosphatases, or PTPs, and ubiquitin ligases. So again, here we have R-kappa B and NF-kappa B. This goes away. NF-kappa B acts in transcription, resulting in pro-inflammatory cytokine release. So, an example of TLR signaling in the action of TLR4, which recognizes LPS. So here we can see a complex of TLR4, MD2, CD14, and LPS is assembled at the macrophage surface. Here's our cell membrane. MyD88, which is a key protein, binds to TLR4 and activates IRAK4 to phosphorylate TRAF6, which leads to the phosphorylation of IKK, or IK. IKK phosphorylates I-kappa B, leading to its degradation and the release of NF-kappa B, as we mentioned earlier, which enters the nucleus. NF-kappa B activates transcription of genes for inflammatory cytokines, which are synthesized in the cytoplasm and secreted via the endoplasmic reticulum. TLR4 uses help from accessory proteins, including MD2 and CD14, to bind to LPS. This signaling induced by TLRs involves sequential activation of protein kinases and adapter proteins. It also involves scaffold formation, ultimately leading to the activation of the transcription factor NF-kappa B. So again, common key steps. TLR ligands bind to TLR. TLRs dimerize, as we see here. They recruit adapter molecules, including MyD88, to the TLR complex via TLR domain interactions. Again, MyD88 is an adapter protein. MyD88 recruits protein kinases, or the IRAKs, and then the IRAKs recruit and activate ubiquitin ligases, including TRAF6 and others, resulting in polyubiquitin chains. This results in, and this ends up with MyD88 and TRAF6, which are adapter proteins, which serves as bridges to bring signaling molecules together. These IRAKs are serine kinases that phosphorylate. So again, summarized here, dimerized TLR4s recruit IRAK1 and 4, activating E3 ubiquitin ligase, TRAF6. Here you can see our TRAF6, IRAK1 and 4, and our ubiquitin ligases. So all those previous steps, as we can see here, we're still going along this pathway, result in the production of a scaffold for activation of a kinase cascade. This recruits adapters and kinases. So we have our adapter proteins, including TABs, or TABs, which bind to the scaffold, and TAC1, which is a kinase, which brings TAC close to IRAK. As we can see here, the result is that the IRAKs phosphorylate TAC1 and cause an active kinase. The active TAC1 phosphorylates IKK, or IK, or the I-kappa B kinase complex. So IKK is a complex of 3 proteins, IKK alpha, beta, and gamma. So we sometimes just call it the IK complex, so it's easier to distinguish between I-kappa. And just so you know, IKKY, or gamma, is also known as NEMO. And so NEMO binds to polyubiquitin chains. So here we can see we got our NEMO binding to the ubiquitin, and we've got our I-kappa alpha and I-kappa beta. The IKK complex, or IK complex, which is also known as the IKB kinase complex, is made up of 3 proteins, alpha, beta, and gamma, or NEMO, which are essential NF-kappa B essential modifiers. And just an easy way to remember finding NEMO. So the activation of IKK by TAC1 will now allow NEMO, or IKK gamma, which binds the polyubiquitin chains and scaffold, which brings IKK complex close to TAC1. TAC1 phosphorylates IKK beta, and IK beta phosphorylates I-kappa B, or IKB, which is the inhibitor for NF-kappa B. Just to clarify, yes, this is I-kappa B. Once phosphorylated I-kappa B, the inhibitory protein, is degraded, and this releases NF-kappa B. NF-kappa B will then enter the nucleus and participate in gene transcription for cytokine genes. The result is expression of genes encoding pro-inflammatory cytokines. Once it is phosphorylated, I-kappa B is targeted for destruction and releases the transcription factor NF-kappa B. Here we can see. Again, also important is that TAC1 also stimulates mitogen-activated protein kinases, or MAPKs, which activate AP1, which is another transcription factor, which is also involved in cytokine expression. But again, this is not shown here. So again, TLR signaling activates the transcription factor NF-kappa B, inducing pro-inflammatory cytokine expression. So here we can just follow along the things we just discussed with TRAF6 and the ubiquitin ligases. Then we have NEMO, polyubiquinated, creating a scaffold. TAC1 associates with IKK and phosphorylates IKK beta, which phosphorylates I-kappa B, releasing NF-kappa B. So these are the main cytokines that you see activated, including IL-8, IL-6, and IL-1 beta, which are all pro-inflammatory. Working together, the TLRs can activate the transcription factors NF-kappa B, AP1, and IRF to induce the expression of inflammatory cytokines and type 1 interferons. So again, not all TLRs use the same adapters and kinases. Some exceptions to the rule are TLR3 and TLR7. So TLR3, like we said before, recognizes double-stranded DNA. Its adapter, it uses TRIF, which is different kinases that phosphorylate different transcription factors. It uses IRF3, which induces type 1 interferon gene expression and antiviral defenses. TLR7 looks at single-stranded RNA, and its adapter, it uses MyD88, but a different kinase activity. The recruitment and phosphorylation and activation of IR7 is used instead, and induces type 1 interferon gene expression, and again, has antiviral defenses. As we can see here in these two figures, it's different from what we saw earlier. We don't see things like NEMO and stuff like that. Slightly different. And these both function in the endosome. So a good summary figure here, just a good graphic. Again, we can see here that there's the 3 transcription factors. They all cross over, but the main one is usually NF-kappa B. Again, we have TLR5, flagellin, and then one of the most common, again, is TLR4 and LPS. So this has come to the end here, but again, I recommend this resource because although it's a lot of detail and I didn't elaborate too much, this is probably the best way to visualize everything. So again, you can follow every step from TLR4, 1, 2, and this is basically the most detailed you get. And if you're really interested in immunology, you can click on each molecule and it can show you what products they have that can detect that, how they measure it. So it's just a good way to, comprehend what they do actually in the field. And even here, you can go from all the way, I want to find MD2, or you can go all the way to the end of you want to find IL-1 beta released in the blood. Simple stuff like that. But yeah, there's so many options and it's super interesting and definitely check it out. Thanks for watching. See you next time.